Mandal Sahib, good morning.
Morning.
Dr. Mandal, should we start or?
Yes, sir. We should start. Dr. Kutub.
Yeah.
We should start now. Okay.
Okay, sure. Okay, good morning, everyone. Respected Dr. Dipankar Maiti, Director, National Rice Research Institute. Today's speaker, Professor Robert Henry from University of Queensland, Australia. All head of the divisions, scientists, and personnel from NRRI and other ICAR institutes. Ladies and gentlemen, a very good morning and warm welcome to all of you. And a very good afternoon to Professor Robert Henry because of the time zone difference. You know, we assembled here to celebrate something very special to us, India's independence. Azadi ka Amrit Mohotsav. Literally, it means the grand celebrations of India's independence. The Mohotsav is a series of events being organized by the government to commemorate the 75th anniversary of India's independence. It will be celebrated as a people's festival in the spirit of people's participation. The official journey of Azadi Ka Amrit Mohotsav started on 12th of March 2021, which starts a 75-week countdown to our 75th anniversary of independence, that is on 15th of August 2022. Science and technology capabilities are fundamental for social and economic progress of a country, especially when it is developing. Therefore, Indian Council of Agricultural Research has taken up a low-cost but people-intensive campaigns on dedicated weekly theme for each of the 75 weeks. Under the ICAR umbrella, National Rice Research Institute is organizing this Azadi Ka Amrit Mohotsav lecture series. Today, we are lucky to have an eminent scientist, Professor Robert Henry, with us to deliver this important lecture. So going directly to the main program, now I would like to request Dr. P.C. Roth, Head Crop Protection Division, NRRI, to formally welcome the guest speaker. Over to you, sir.
Good afternoon, Professor Robert Henry and colleagues in Australia. Good afternoon, good morning, everybody in India. I take this opportunity to extend a warm welcome to Honorable Robert Henry, Professor of Innovation in Agriculture, Queensland Alliance for Agriculture and Food Innovation, the University of Queensland, Australia, to our institute through virtual mode on the occasion of a special talk series to celebrate the 75 years of India's independence. I welcome our Honorable Director, Dr. Dipankar Maiti, to preside over the virtual function. Hearty welcome to all the dignitaries of ICAR headquarters and other ICAR institutes, head of divisions, and scientists of ICAR and NRRI, technical staffs, students, project staff of NRRI to this virtual function. Sir, we are celebrating the Azadi Ka Amrit Mohotsav on the above 75th year of India's independence from August 2021 till August 2022 by organizing special talk series. In this regard, we organized first special talk on 18th August 2021 on topic agripreneurship-led extension by renowned scientist Dr. P. Chandrasekhar, Director General, National Institute of Agriculture Extension Management, Hyderabad. Today, this is the second lecture in the series of special talks to celebrate this 75 year of independence Azadi ka Amrit Mohotsav. Genetic potential enhancement and development of value chains to rice is one of our focus area for research and development. Genomics study of domesticated and wild rice relatives highlights genetic conservation, turnovers, and innovation across the genus Arizer. Professor Robert Henry leads the way in research into genomic sequencing to capture novel genetic resources for the diversification of food crops, including rice, to deliver improved food products. He has vast experience in wild rice germplasm, particularly with the Arizer australiensis. Professor Henry has kindly accepted our invitation to deliver a special talk on genomics of wild and domesticated rice in this virtual platform. I firmly believe this lecture will serve as a source of knowledge and strategy for the scientific community working on rice and allied crops. I welcome you all to enjoy the special talk by Professor Henry. Thank you.
Thank you very much, sir. Although most of us know Professor Henry and his great contribution, for many participants from other institutes and students of this institute, I request Dr. Sudhamai Mandal, Principal Scientist at NRI, to introduce Professor Robert Henry to a broader audience.
Thank you, Dr. Kutub. Good afternoon to Professor Robert Henry and colleagues in Australia, and good morning to everybody in the august audience in India. It's my proud privilege to grab this opportunity to introduce the guest speaker to this august gathering on this virtual platform. Professor Robert Henry conducts research on the development of new products from plants. He is particularly interested in Australian flora and plants of economic and social importance. His research uses genome sequencing to capture novel genetic resources to improve understanding of the molecular basis of the quality of products produced from plants, and genome analysis to capture novel genetic resources for diversification of food and energy crops. Professor Robert Henry is basically a professor of innovation in agriculture and was foundation director of the Queensland Alliance for Agriculture and Food Innovation, a research institute at the University of Queensland, formed in partnership with the Queensland government. He was previously research director of the Grain Foods Cooperative Research Centre. He was director of the Centre for Plant Conservation Genetics at Southern Cross University in Australia. He was research program leader in the Queensland Agricultural Biotechnology Centre also. He has been involved in establishing several cooperative research centres in Australia and has contributed to the management of research funding by rural research and development corporations in Australia. Coming to his academics, Professor Henry is a graduate of the University of Queensland, BSc Honours, Macquarie University, MSc Honours, and La Trobe University, PhD. He was awarded a higher doctorate, DSc, by the University of Queensland for his outstanding work on the variation in plants. He is a fellow of the Royal Australian Chemical Institute. He is the recipient of the prestigious Guthrie Medal for his contributions to cereal chemistry. He is a fellow of the Australian Academy of Technological Sciences and Engineering. This is, in brief, his salient career in his life. Thank you, sir.
Now it's time to listen to Professor Robert Henry. So it's over to you, sir, for your lecture.
Thank you very much. Thank you for the kind introduction, and thanks very much for the invitation to speak to you today to help to celebrate such an important milestone. I'll now look to share my screen. Hopefully. Can you see the screen now?
Yes, sir.
Says good. Thank you. So I'm going to talk about the genomics of wild and domesticated rice, and I might start by thinking about the future of agriculture and food production, and of course rice's place in that. And really important issue for us today is that agriculture needs to be adapted to climate change, and rice is very much part of that adaptation. But at the same time that we're aiming to adapt our agriculture to climate change, we need to continue to address the many demands that we have. We need to continue to produce foods that meet consumers' requirements for taste and convenience. But increasingly, consumers globally are asking more. They're expecting foods to be healthy, sustainably produced, and even ethically produced. And these are all new demands being placed on food that we need to meet with.
Could you please
We okay? Speaking? Yes, thank you. The real responses that we have the opportunity to make to climate, of course, are to adapt, and that means, I think, with rice, a lot of genetics to produce rices that are adapted to new environments. We can move to agriculture, and we are seeing, particularly in Australia, the opportunity to move rice quite radically from temperate areas into tropical production zones. And that, of course, also requires substantial genetics, because we are going into a new environment, even though we're moving to a more suitable environment as the climate changes. Or the other more radical option that's being adopted with some crops, high-value crops, is to protect them. Now, in the case of rice, this is only going to happen for extremely highly valuable varieties, but in general in agriculture, this means really quite radical genetic inputs, because instead of adapting our crops to a variable environment, we now have to adapt them to their best performance under a specialised controlled environment. And this may be a path we go down for rice, particularly for specialty rices. I'd like to turn to an example of the challenge we have with climate resilience in rice by talking about fragrant rice. And many years ago in my laboratory, we identified the gene that controls fragrance in rice by getting access very early to the first examples of the rice genome. And because we had markers flanking the fragrance gene, we were able to identify it very quickly, and by expressing it in bacteria, we could understand the chemistry associated with this. And immediately we discovered that fragrance in rice is associated with a loss of a function of a gene that's actually important for the performance of the plant under stress. This is the pathway of proline metabolism that's upregulated in the plant in response to abiotic stresses, salt stress, drought stress, and so forth. And so fragrant rices are not as fit in their essential characteristics as non-fragrant rices in these environments. So we've been using genomic approaches to try and understand how we can compensate for that and produce climate resilient fragrant rices that will perform well under stress conditions. And just to illustrate how widely we can look in our genomics searches, I'd like to just refer to some work we've done in collaboration with Dr. Nadaf at Pune. Here we've been working with the fragrance from pandan, and this is exactly the same chemistry as we have in rice, and essentially the same genetics. And what we've been able to identify by looking at the genes here is different pathways of metabolism that allow this plant to be both tolerant to salt and to be fragrant. And so by learning from these species, we can think about the sort of metabolism that we need to put into a rice plant to make a particularly stress-tolerant fragrant rice. So genomics can allow us to move across species to make these discoveries. So I'd like to talk much more generally about the plant genetic resources we have and how we can better utilize them using genomics tools, and particularly using the example of rice. But in working with a wide range of plant genetic resources, we need to pay attention to the issues around access and benefit sharing associated with the utilization of those resources. And we are in an increasingly complex environment internationally. I've been working with our law faculty to understand the implications of applying Nagoya protocols to access and benefit sharing for a wide range of agricultural species. Rice is a special case because it's dealt with in the plant treaty, but nonetheless making sure that we are dealing with our germplasm in a Nagoya compliant way is a challenge, because each country has a different interpretation of Nagoya and different rules and ways of considering when Nagoya started, whether it's historical or begins at a particular point in time. And so we really do need to continue to collaborate internationally to develop a framework that allows us to access the diversity of resources we need for rice and other crops. So in capturing biodiversity, we can look really across the whole plant kingdom. And for much of the work that's been done in my lab, we really have ranged far and wide in finding the genes that might be of use to us. If we turn back to the rice group, the Arizer and related genera really do have quite a lot of diversity that we're yet to fully exploit in domesticated rice. So we still have plenty of material within that gene pool worthy of further utilization. So I'm going to talk about applying genomics, and I'll just show these books that I've been associated with over the years that have described the sort of technology as it's evolved for applying molecular tools in plant improvement. And it's fair to say that the technology continues to develop at a very rapid rate, and that really the trend has been towards much more complete analysis now. So instead of looking at genes, we very much look at genomes. And so the best approach is not probably to look at individual molecular markers, but now or very soon in the near future will be to look at the sequence of the whole genome of any individual that we're interested in. The cost of doing that and the richness of data that it provides makes this an attractive option for the future of the way we analyze the genetics of any of our plants. And this is being driven really by the continued collapse in the price of sequencing. Really the cost of sequencing plant genomes gets smaller all the time. And in the case of rice, of course, because we have such a high quality reference genome, we're really resequencing, and the cost of resequencing is very low to get really a fairly good analysis of every base in the genome. And so for very large collections of rice now, we can begin to look at the whole genome and don't have to make any decisions about specific genes. And a lot of this, of course, means that our research is moving effectively in silico. I wrote this piece a little while ago on how plant science was escaping from the laboratory and really was moving into the computer. And I think that increasingly that's the case, that much more of our work now is at the keyboard rather than at the laboratory bench in working with genetics of any organism. But rice is certainly a good example of this movement to a very large big data environment in which we conduct our research. And the plant genome analysis is contributing to most importantly, to the biological understanding of the organism that's key to genetic improvement. If we really understand the molecular basis of the trait, then we're in a much stronger position to manipulate that trait and to select for the attributes that we want. We really work in my team at three sort of levels. We work with chloroplasts, and here we're able to look very much at reticulate evolution in many plant groups, including the Arizer. We're working very intensely with transcriptomes, and here this is a key tool for gene discovery, comparing the genes expressed in different genotypes or at different stages of development, or in genotypes with different phenotypic characteristics. And increasingly, we're now focusing on the issue of allele-specific expression and how that can explain phenotypic variation. But in the nuclear genome, this really is where we do most of our gene discovery, by comparing the genes in individuals with different phenotypes. But increasingly also, we're looking at the importance of heterozygosity at each locus, and in the wider gene pool, in the presence and absence of genes in different individuals that may explain a large part of the genetic differences that we see. To do this, we've undertaken some recent comparisons of all of the available technology, the various sequencing platforms that are available for us to use, the Oxford Nanopore technology, the PacBio technology, and the BGI long-read technology. These are three different ways in which we can get long DNA sequences, which are very powerful in putting together de novo genome sequences. But we're also, of course, working with the traditional short-read Illumina sequencing and using techniques like Hi-seq to put together chromosome-level assemblies of our genomes. This analysis is allowing us to chart the improvements in sequencing by applying each of the techniques to the same samples and comparing both the chemistry advances and also the software advances in analysing the sequence data that we get back from our instruments. And we're seeing a real improvement in both of those areas, particularly in the last 1 or 2 years, that really have driven us to a new level of capability in de novo genome sequencing. And I illustrate here in these examples for this plant where using the PacBio HiFi method, which repeatedly sequences the same molecule to give us very accurate long reads, that we can assemble the whole chromosome to a single COMTIB just by assembly of the sequence data. We need no other information. So it's true that for most chromosomes in plants, we get the whole chromosome now directly from our sequence data without any other prior knowledge. And that's a really big advance over where we were a year or two ago in being able to put together de novo genomes of many different individuals. So in working with rice, this editorial a few years ago described what we were doing as reinventing rice. And I think we have to continually, in all of our crops, reinvent them to suit the new circumstances we find, the new pathogens, the new disease environments, the new environmental constraints, the new consumer expectations of the food product. The rice, of course, is very much part of a pan-tropical genus, the Arizer, and working with many groups internationally, it's been possible to put together the genomes of most of the species in such a way that we can really begin to understand the evolution of this group and the evolutionary relationships. And this has involved movement of rice around the world, its divergence in different geographical locations, and then various introgression events and further translocations and movements at different periods of time. So a complex history that you'd expect from a species like this. Much of our work has been in the what we call the AA genome species, which are the close relatives of domesticated rice, the species with which rice is to a significant extent interfertile This represents the primary gene pool of rice. And we see divergence of an Australian species, Arizer meridionalis, about 2 to 3 million years ago from the group that we see in Asia and Africa that led ultimately to the domestication of rice. So this is a group that contains a lot of genetics that's potentially useful for rice. And we have also in Australia something called Arizer roofy pogon that's actually distinct from the Asian roofy pogon and probably diverged about 1.5 million years ago. And so this is another sister to the domesticated clade. And I'll come a little bit to talk about that soon. So this allows us to see that we've got this Asian and African clades from which rice was domesticated, and as a sister to that, we've got the Australian group, the group of wild relatives there, and then outside that, various South American, African rices, and other Australian rices like Arizer australiensis. And to explain this a little bit more, what we see here is that this is a phylogeny based on analysis of about 4,000 genes, that we have this Australian clade that diverged back about 3 million years ago from the clade leading to domesticated rice, and then another Australian group diverging at about 1.7 million years ago. That's Arizer roofy pogon in Australia. It's very morphologically very similar to roofy pogon in Asia, but it has a chloroplast that puts it in the clade with meridionalis. So this is somewhat intermediate between meridionalis and Asian roofy pogon. And then we see the subsequent divergence of Asian and African gene pools and the domestication of Japonica and Indica rices. So just to explain that a little bit more, the variety on the extreme right, Meridionalis, is the most divergent of the A genome species. Then we have the one in the middle, the Australian roofy pogon type, diverged about 1.5 million years ago. And these are very important parts of the primary gene pool. They're interfertile with domesticated rice. They have novel resistances to pests and diseases that have evolved in the Australian landscape. And they also have novel health and nutritional traits that we're looking to move into the domesticated gene pool. So these represent the greatest diversity that we can find within that primary gene pool. Just by comparison, on the left is Arizer australiensis, a much more earlier diverged form of rice, a very tall plant with a short grain, whereas the others are medium long grain. And we see in Northern Australia very large populations of wild rice. In many places, it's the major understory. Wherever it's wet, we see rice and of course, emergent trees. And these populations extend right across Northern Australia for thousands of kilometres. So this is a summary of the species that we find in the tribe. Quite a lot of species at varying evolutionary distances from domesticated rice. We have this species, Photomophila parviflora, which I think should be of interest to rice geneticists. It's a genus only found here in these rivers on the east coast of Australia. Its closest relatives are probably the zizanias of North America. And the novel feature here is that it has separate male and female flowers and relatively small flowers and seeds. So Photomophila has got some cold tolerance in that it grows in quite cold environments in the mountains, and so represents a source of cold tolerance and this novel attribute of single-sex flowers. The Lursia is another very distant relative that's really quite widespread. I'm not sure we've identified what traits we have in that. We do have Arizer officinalis in Australia, but it's very limited in distribution, found on an island in Torres Strait. I've been traveling to that island to try and find more resources for it, but very limited recent collections, and we haven't been able to find this species in the wild in the last decade or so. But of course, Arizer australiensis, very widespread across northern Australia, and Arizer australiensis is quite novel in that it has a rhizome. So in the dry season, it dies back to that rhizome, and when the wet season comes, it shoots back again. A very novel lifestyle for a rice. It also has novel starch properties with low retrogradation, and it has novel photosynthetic capacity, a novel rubisco gene that is more efficient than that in other Arizer. So a real source of novel and important genetics. We think a source of nutrient use efficiency for nitrogen and phosphorus that might be able to be captured for rice improvement as well. Then we come to Arizer roofy pogon, and morphologically very similar to Arizer roofy pogon in Asia, but as I've indicated, the sequence shows that in the nuclear genome, it's a sister group to the Asian group, and the chloroplast genome actually is much closer to what we see in Meridianalis. And here Meridianalis with the closed panicle is a species that's very widespread. This is the one that has very large populations and is found only in Australia, but has recently been seen in Papua New Guinea, I think, so it may extend across Torres Strait. When we look at the chloroplasts in our wild rices, we look at those in the region, and the region that we look at is from northern Australia to northern China and across to India, and there we see quite a number of different clades of wild rice. Interestingly, the Australian chloroplast type we found in the southern Philippines. And that's not unusual that we see exchanges of flora between those regions, it's quite likely that there's dispersal by birds or other mechanisms between those locations. What we've done is focus on looking at many of the wild populations in remote areas where there's been little interaction and sequencing these wild rice populations. And what we've found is that there is the existence of hybrids in the wild. So we're seeing actual evidence of reticulate evolution in Arizer, where we see two species forming rarely individuals that have intermediate morphology, and when we analyze the sequence, we find that they're actually hybrids. So we've got two quite distinct species with some sort of reproductive barriers, but a low level of introgression happening. And this is, I think, an indication of the history of rice globally, that this has been part of the process where we have divergent species, but we have the possibility for gene flow between species at a low level, which is an important element of the way these species have evolved. We do also find very large grain size, and this is if we look at an Asian rice there at E, we have B is Meridianalis, and we can see here, because of differences in grain size genes in these wild populations, that we have a predominance of large grains in the wild, larger than we would see in a domesticated rice. And this shows us that we've got useful genes for that. And we can back cross this into, working with Japanese colleagues, we've been able to back cross this grain size gene into a domesticated background, showing that we get maybe that 20% increase in grain size with this single locus from the wild rice. I'd like to say a little bit about what we've learnt with chloroplast sequencing in rice. This is a technique that's dramatically improved by using next generation sequencing. We now don't have the problem of contamination or confusion with mitochondrial and nuclear insertions of the chloroplast. We get the genuine plastid genome, and we've improved dramatically the annotation with the latest software. So we now get all of the genes, we get the complete base perfect chloroplast from every sample we've sequenced, and we get full annotation of all of the chloroplast genes. And by looking at that, we're able to look at relationships between rice populations and study both wild and domesticated rice. In the case of domesticated rice, we find two clades, and many people would say, well, that's what we'd expect. But you'll see these two clades here have some outgroups, and they're the wild Australian and Asian and African clades. But the two domesticated clades are quite remarkable in that they have quite a bit of diversity within them, but none of it is functional. So there's actually only two haplotypes in domesticated rice, and they're not indica and japonica. Indica and japonica have exchanged chloroplasts such that we see indica and japonica and all the other subtypes of rice within each of these chloroplast groups. The two clades differ in many important genes. They differ in function in the amino acid sequence of a number of photosynthetic genes. They differ in the sequence of even ribosomal genes. So there's quite a number of functional differences, but only two functional haplotypes here. So what this tells us is that rice has only two mothers, but many fathers. And that's the simple way of thinking about it. It's clear that there was only two chloroplast types, functionally distinct types, that were captured in two separate domestication events. Maybe one was in India, one was in Southeast Asia or China, but those rices have moved around the world and incorporated genes from local wild rices to form the 5 or 6 distinct types of rice groups that we see in a nuclear phylogeny, but with only these two maternal genomes. So this discovery really suggests that we have the opportunity to bring new maternal genomes into the rice gene pool, and that they may actually provide some advantage in different environments, because these two chloroplast types may provide adaptation to different environments. For example, different light intensities, given that there's such significant differences in some of the photosynthetic genes. Rice also has the potential to have new starch properties by working with the wild material. We've been looking at the digestibility and finding very low digestibility that's desirable in rice from a human health perspective in many of these wild rices. And what's unique about them is that we can bring together our understanding of starch properties and starch chemistry and the genetics of rice starch to think about how we could breed rices with starches that would be healthier. Now, for a long time, it's been possible to produce rices with lower starch digestibility, lower GI, for example. But many of these rices have been unattractive. The texture has been undesirable. But what we now see by doing a lot of sensory evaluation is that we believe by using some of the great diversity we have in the wild, we can create rices that are both healthy, have low digestibility, a low glycemic index, but also have the attractive tastes and textures that the rice consumer is looking for. And so this is an important use of wild diversity where natural selection has produced some of the variants we want that haven't been possible by just conventional mutagenesis. So I'd just like to say a little bit about our local experience in trying to develop a rice industry in northern Australia, where we don't really have one. Our rice industry is in the temperate areas where we grow rice on irrigation water, effectively in the desert. Climate change means that water is becoming much more difficult to assign to rice because of the demands of perennial crops. We need to move our rice dramatically into the tropics, where we've got rain-fed environments, and here we're going to need to borrow much of the genetics and experience of the tropical world in developing new varieties for these new areas. And we've been doing this in association with our Cooperative Extension Centre for Developing Northern Australia. And these, of course, can build on the fact that rice is very much a native plant in these northern environments, but we have new challenges. We have our own strains of blast and other rice diseases. Because rice has been evolving for millions of years in the wild in Australia, so have the pathogens. and we've got different strains of the pathogens than you would find in Asia. So this explains to some extent why we bring Asian rices into Northern Australia. We get serious disease problems because we don't necessarily have resistance to the natural pests and diseases. Whereas, as we've done in the past, growing rice in the south, we grow rice in an environment where there is no natural rice, in a colder environment where, in fact, we don't have any rice diseases. So we're going to have to confront the challenge of breeding rice with disease resistance for a new range of pathogens to establish a rice industry in response to climate change in the places where we've actually got the water. And of course, we want to take the opportunity in going down this path of capturing the grain size and colour and starch properties and many of the other desirable attributes that we can get from the wild rices, and of course the disease resistance. We believe we can back cross resistance from the wild into domesticated rices to provide rices that will be suitable for those environments. So we're looking at our ability to undertake domestication in rice, and the way that we're approaching that is to look at doing that by gene editing with CRISPR-Cas systems. So we're developing routine and high throughput systems for both wild and domesticated rices so that we can actually take any gene we find in a wild rice and edit it into a domesticated rice, or we can take any domestication gene from a domesticated rice and edit it into a wild rice. And so we can quickly evaluate the phenotypes of any of the genes we find in the wild gene pool using the newer gene editing approaches. And so there's been a history of rice in the north, but it's not been successful because we haven't confronted the disease problems, but we are now about to do that. So we're looking at three types of options. We're looking at an industry where we cultivate some of the wild rices as novel products. We're looking at domesticated Asian rices, and we're particularly looking at unique styles of rice for the region that incorporate some of the disease resistance from the wild. Just like to say a few words about what we're doing some other species that have real relevance to rice. We're also working on sorghum, and sorghum is another tropical species, another cereal that has many characteristics that might, in more radical sense, be useful to understand from a rice perspective. And so we're working with sequencing all the genomes of the many species of sorghum, and they're mainly found in Northern Australia, despite sorghum being domesticated in Africa. We have a new project on tackling the challenges of climate change by producing a massive population of mutated sorghum. So we're on the path, and here's the first samples in the field, of producing 500,000 different lines of sorghum, each with mutations produced by chemical mutagenesis. So objective will be to have a mutant in every gene available off the shelf, characterized in sorghum. And this is being conducted by high throughput screening, robotic type screening capability in Denmark. What this allows to do is things like the work that we did with Dr. Nadaf on looking at the fragrance in rice in sorghum. Because sorghum, if we knock out the same gene, is also fragrant. So we can look at fragrance, and we can look at some of the issues around plant performance in fragrant and non-fragrant sorghums, and understand how we can produce fragrant cereals that have the right stress tolerance and so forth. So we're beginning to look at all the gene networks in sorghum and which ones of those might actually be useful if we deployed them in a rice background. We're also looking in these systems at allele-specific PCR, and here this is a diversion out to just explain that by looking at sugarcane, which is effectively a polyploid sorghum because it's got 12 copies of all the genes, we've been able to relate across the whole genome gene expression to copy number, and that's something we can come back to in rice. We can look at the effect of allele-specific expression in the diploid rice by doing that. Here, remarkably, we've been able to resolve 1 copy from 2 copies from 3 copies in the genome and show that we get for many genes, unbiased expression relative to the dosage. So if it's heterozygous or homozygous, we would get 1 or 2 doses of expression, but that we do see genes that show completely biased expression with much stronger expression of one allele. And that's something we're trying to understand now back in the rice system. So what we're really aiming to do is characterize the domestication genes in wild relatives of rice. And here's another example, a more extreme one, Microlinus dipoides. We're looking at this. This is a temperate grass that can grow in quite dry areas. And it has a grain that we can now get up to being similar to rice sizes. It's in the Arizer tribe, and it represents an opportunity for a dryland crop that can produce a grain that could be consumed in the same way that we consume rice. So here's an opportunity that we've been able to show that by mutating the shattering genes in this, we can get a non-shattering version. So we're beginning the path of domestication of these species, and domestication now can be very rapid because we can do it in 1 or 2 generations, because we can edit or mutate any of the genes we want to put in all the things that would take a wild plant to being one that would behave well in a domesticated environment. And so this can be applied not only to domesticating, of course, crop wild relatives, but to domesticating things that are right outside that range, that have never been crops before, what we consider completely novel or new crops. And some of those could be targeted at a whole grain product, at a rice type product, rather than what most cereals are used as something that can be ground up and used as a powder. Rice is quite unique in being consumed as a whole grain product. Finding other species that can fill that niche is challenging, but worthwhile, particularly for environments outside the range where we could grow rice. So what we're doing is characterizing the genomes now of all our wild material so that we can identify all the target genes for manipulation in going through this transition to domestication. What we're doing is also, I'd like to explain, undertaking a major effort in working with the Genomics of Australian Plants consortium, where we're aiming ultimately to sequence all plants in Australia. We have this long-term ambition now, of course, to sequence all organisms on the planet, and systematically in plants, the approach that we're now taking is sequence all families of plants, then at the second level, each genus, and we're now working through all the genera of plants, and then we come down and attempt to sequence all of the species. Now, to do this globally is a big effort, but I think it will happen faster than any of us imagine, because the rate at which the technology is advancing. And of course, we can also use genomics to understand environmental interaction. I go back to a study we did many years ago where we looked at the genome of plants growing across an environmental gradient here from a moist slope to a dry one, and asked the question, under natural selection, how do plants adapt to different environments? Can we learn from that how plants can be adapted in agriculture to new environments in the future, particularly if the climate changes? And here's another example where we've worked with, this is with the Microlina, the Arizer tribe species, from Melbourne up over the mountains and back to sea level through a wide range of temperatures. So here we can explore how the plants have adapted to extreme temperatures, hot or cold. And what we find here, as in other studies, is that the main adaptation of these plants in the wild to temperature differences is in fact seen in their resistance to pests and diseases. It's the increased biotic stress that the plants suffer at higher temperatures that is the main challenge, rather than the temperature per se. And I think this is true for agriculture. So in adapting to higher temperatures, we need to adapt to the pests and diseases the plant will encounter in those environments, rather than the temperature per se, because quite often the plant can cope with that. And so we really need to take those learnings in the way we approach developing climate resilient crops, including rice, for the future. So I believe that rice represents a great example of how we can work with crop wild relatives to improve traditional rice, but also to think about some quite new or radically different crops that might be key solutions, particularly in extending the range of environments in which we can grow foods that are consumed in the same way as rice. In my team, we're working across quite a range of species: rice, sorghum, pigeon pea, macadamia, eucalypts, and citrus. In all cases, we've got extensive wild material that we're using to undertake this interaction between the wild and domesticated groupings. And I'd just like to explain a study that I was involved with a number of years ago that really takes us back to take a broad perspective on this. This is wild barley here in Syria, and this is the first plant that we believe that humans domesticated 10 or 12,000 years ago. And there's been many mysteries, one of which has been the origin of the wild barleys or the domesticated barley in Tibet, and there was a thought there was a separate domestication there. Well, sequencing large numbers of genotypes has given us an answer to that, as it will to many of the questions we're now asking. And what it's shown is that this barley was in fact domesticated in the Fertile Crescent in the eastern Mediterranean, but then taken across through India and up into the Himalayas in more than 5,000 years ago. And it went through a genetic bottleneck into those colder climates because very few genotypes survived in the cold. But then, remarkably, it escaped into the wild. So we now understand that the wild Tibetan barley was not the source of the domesticate, but really an escape into the wild. And genomics can be really important in unraveling the history of crops, and that's so important in charting the path for the future of our crops. Here in Australia, we've had 65,000 years of continuous human habitation across most of the continent, and food production has been undertaken from very large number of plant species, but the knowledge about this is frequently lost. So we're going back now and looking at many of the wild populations to see if many of these species were potentially food crops in the past that may have been passed by or may have escaped. And we're looking for signatures of human selection, and we're finding some species that seem to have genetics that does suggest that they were subject to widespread human harvesting and use in the past. So this genomics allows us not only to go forward, but also to go back and to learn from that as well. One of the big challenges is finding a way we can redesign plants for non-food roles, and that's particularly in replacing fossil carbon. We need to replace fossil carbon with renewable carbon, and plants is where that's got to come from for all of the things that we make from oil, like our plastics and chemicals and so forth. And so we need some fairly radical genomics and redesign. Now, rice is not necessarily the primary crop here, but the biomass of rice may well contribute substantially to non-food products in the future. And so I think it is relevant to think about how we use the rest of the plant, not just how we use the grain. And of course, this would be a way to avoid, I think, burning of rice stubble if we could find a way of utilising it for high-value products. I finished by saying we're going to have a symposium here in Brisbane, the Harlan Symposium. We'll talk about crop domestication, and that will be part of our TropAg conference to be held late next year. We're hoping that COVID's going to allow people to travel and that we won't have to do everything online. But I'd then just also like to acknowledge all of the many sponsors of the research that we do, and of course thank the many collaborators that are involved in the research that I've talked about today. So I'll finish there and be very happy to take questions. Thank you very much.
Thank you very much, Professor Robert Henry, for the lecture. So it was a great learning experience for all of us. You know, starting from almost, I think you touched almost everything regarding the wild relatives of rice and how they could be utilized to improve our crop varieties in the changing climatic scenario, starting from nutrition to stress adaptation. Also, you touched on, a little bit of sorghum domestication also. So when you did not start on talking about de novo domestication, So I prepared this question for you because it came to my mind. So what do you think about which wild relatives of rice is the most suitable one for de novo domestication using genome editing technology?
Well, I think it must depend on what the end product that you're looking for is. I think it's possible to think about it with any of the rices that might have a grain that is of a type suitable for human consumption as a food. And that would include many of the species. So it really depends on whether those species offer particular production advantages in the environment that you are going to domesticate it for. But I think all of them are possible, but I think of course the A genome species are particularly suitable because they're so close. We really can think about bringing any of the major traits we've got in rice into those, because there's probably a relatively small number of genes we'd have to work with to do that. And I do think that some fresh domestications bringing in a new diversity would be very useful for rice breeding in general. And using the new technology we've got, being able to sequence and understand exactly what we've done and set our goals. I think there's many options. I'm looking here in Australia at our local species as something to domesticate for local production, but that might be the principle that applies elsewhere, that it may be the local wild rices that are the best option for domestication in that region, because they may contain local adaptations that are useful to capture.
Okay. Thank you. So actually it is the time for discussion from the participants. They can directly ask you a question. So before going to the participants directly, I have another simple question for you. So you talked about a lot about sequencing technology, so about Oxford nanopore technology also. So I heard that it is very small, and it can be it is very much good for portable use. So how difficult is it to analyze data generated by Oxford nanopore sequencing? Means how much technical expertise is needed if we want to buy that?
So you're right. I think we've overcome the technical barriers to some extent in terms of the chemistry, and we've got portable devices, low cost devices. We can get a lot of data very quickly, but quite often it's the data analysis that's the challenge. And we're expecting to see more advances in the software all the time. The software now is as limiting as the chemistry in getting to better genomes. We do find at the moment that the Oxford nanopore data is cheap to produce, but quite challenging to analyze, because you've got large data volumes of sequence that's got still quite a lot of errors in it. The big advance with the PacBio platform really has been the movement to greater accuracy. That greater accuracy has actually helped with the data analysis, because if you've got long, accurate sequences, it's very simple to assemble them. You don't have the challenge of the software having to do a lot more work to get the genome together. So the time it takes, the computational time it takes to assemble a genome from PacBio data, for example, has completely collapsed in the last year or so. From taking us these long read sequencing assemblies. Now, I can remember when we typically expected to take 6 weeks or 8 weeks to do a computation at a standard sort of computational rate. Now we expect those things to happen in hours. And that's largely because the starting material that the computation's based on is more accurate sequence. Now, I understand that Oxford nanopore is undergoing a major chemistry upgrade that will improve the accuracy of the reads. That will hopefully reduce the computational time. And so it won't just be you're getting more accurate sequence, you can actually deal with it more readily. So that's the innovation we're looking for.
Okay, thank you. Hope it will be simpler soon. So yeah, so now I would go to Dr. Mohesh Yadav, uh, principal scientist from ICAR. So you can unmute, sir, and you can directly ask this question.
Good morning. Thank you very much, Dr. Fatbuddin. My question is to Dr. Henry that the Oryza meridionalis, based on the chloroplast sequencing, how much it is different from the Oryza nevara?
It's quite unrelated to Nevara. Nevara is much closer, of course, to roofy pogon, and yet it's quite distinct from roofy pogon. So I can't remember the numbers, but it's quite substantially different. It would be many times the genetic distance that there is between Nevara and roofy pogon. So it genuinely is in that if we think of it in molecular clock terms about the chloroplast sequence, that Meridianalis diverged from the other clades about 3 million years ago, whereas Nevara and roofy pogon in Asia have diverged probably less than 1 million years ago. So many times further away.
But both are the annual and growth habit. So beside this chloroplast genome, how much similarity between the meridionalis and Oryza and nevara about the nuclear genome?
The nuclear genome is here that we don't see any sort of reticular evolution. The nuclear genome shows a similar sort of divergence. Where we see a discordance between chloroplast and nuclear phylogenies would be in the Australian roofy pogon, where the nuclear genome shows close relationship with Asian roofy pogon, and the chloroplast genome shows close relationship with the Meridianalis. And that suggests chloroplast exchange has occurred within the Australian environment between the two species at some time in the last sort of 1 million years or so.
Sir, based on the chloroplast genome, do you think that Oryza, this particularly Brachyantha, that was based on the different studies of cytogenetics and molecular genetics, was considered as on the periphery of Oryza genus? Based on the chloroplast genome, what was your take, sir?
I'm not sure which taxa are you interested in?
Oryza Brachyantha, F genome. Oryza Brachyantha.
Yeah, so I'm not familiar with that data. I just caution that the nuclear and chloroplast phylogenies are not consistent across the whole genus. The other great example is the South American and African case where it seems that chloroplast has moved across the Atlantic Ocean in relatively recent times, because we've got species on either side, nuclear genomes quite distinct, but apparently sharing a much closer chloroplast. So it sort of comes to the point I made where we actually are able to find these rare hybrids in the wild that show that these species are hybridizing, and that means the chloroplast moves into the other population, and so that it can happen at a very low frequency, and so we see that in many plant groups that are closely related, that there's a small level of interspecies, despite the species being distinct, there's a small level of gene flow, and that quite often means the chloroplasts move or exchanged in a way that means the chloroplast phylogeny and the nuclear phylogeny just don't match. And that's the real result, because that tells us that the evolutionary history of the chloroplast and the nuclear genome follows a slightly different path in closely related species.
My last question, that Oryza correctata, earlier classified as a different species in a different genera. Now it's Oryza in the Oryza genus. Do you think
Yes.
the Oryza correctata and Oryza this Brachyantha, which is more closer to the species of genus Oryza, like different polyploid species are there. This is of course also a polyploid species.
Again, I haven't looked at the relationships within that group, but I think it is reasonable that this is back in the genus. And of course, polyploidy is not a big change. We find polyploid differences within species sometimes. So I don't count one being polyploid and the other not as evidence of a big genetic difference, because that can happen fairly quickly. So I think I think the understanding is moving in the right direction, but I haven't focused on that.
Okay, thank you very much. Thank you.
Yeah, thank you. A lot of questions. So now we have to move to another participant, Dr. Yesing, if I am not wrong in pronunciation your name. Dr. Yesing, please unmute yourself.
Hello, Professor Henry. Good afternoon from India. Actually, you said like you are working on wild relatives of pigeon pea also. What kind of trait you are looking for?
Sorry, can you repeat the question? I didn't.
Pigeon pea?
Pigeon pea. Yes. So in pigeon pea, the wild relatives here have resistance to insects, and of course insects are a major pest in our environment. Because again, it's a native species, we have a lot of insects that seem to cause very severe damage in our pigeon pea, and we have strong resistance in the wild relatives, and we've been successfully able to back cross that into pigeon pea, and we're now using the genome, of course, to help it to accelerate that back crossing of insect resistance.
In case if you are crossing that wild relative with a cultivated pigeon pea, you will be getting some of the undesirable traits also along with that. Like our wild pigeon peas are not edible one. So
yes.
How we are planning for the future, like after crossing it, it is only for the study purpose or to develop something which is edible for the common public?
So it requires that we have a molecular understanding that allows us to do repeated backcrossing. That's the strategy, so that we can backcross until we get rid of all those undesirable traits but retain the insect resistance. And of course, that's going to be much more efficient when we have the molecular markers for the genes controlling the insect resistance. You're right, the early generations will have all sorts of undesirable traits coming from the wild. That's the same with rice, of course. Yeah.
Thank you.
Okay. Thank you, ma'am, Dr. Yasin, ma'am. Yeah. So now, Dr. Somnath Rai, you please go ahead.
Yeah, Professor Robert, excellent presentation. Actually, I just want to know your opinion about the de-domestication in weedy rice. Is it really the reverse process of domestication?
Probably. I think that's the other example I talked about of de-domestication was in the wild barleys, of course, and that's we understand it quite well because we've really looked at the sequence of the shattering locus in across the wild and domesticated gene pools and really pieced together the history of that. I think that the weedy rice must be considered in that same category as a mutation going the other direction, even though that clearly seems to be a less likely event in many ways because of what the change that needs to happen.
Okay, thank you.
Thanks. So next, Dr. Middle Chakraborty, please.
My question is regarding Orizer nivara. This Orizer map alignment project, you were also one of the team members. So in Orizer nivara, only one accession has been sequenced and given. In all the cases, only one accession. But sir, my work experience with Orizer nivara, I am doing quite detailed work here. If you at least 5-6 accessions, of, at least in case of nivara, it is not done. The consensus sequencing that cannot be developed, and my experimental evidences suggest that there is among the A genome species, maximum transposable element related movement is in the Nivara species. Whereas the Nivara, based on your sequence available, and I have compared those things. Some part I have already published in Euphytica recently, also reported it, and some more detailed results are going to be published. So and another said that from your Arizer map alignment group, that project group, Dr. Oliveira from Brazil, his group regarding the subart genes evolution, they have reported something like it is present in chromosome 1 and this kind of thing. And we have got very well evidence that that is also not stable. Actually, in different accessions of Nivara also, that movement, there is movement of that loci. So in that respect, sir, I also want to know your opinion that to get a standard sequence of Arizer Nivara, at least Indian Nivara and India also, like Odisha, Odisha provincial accession should be sequenced. Otherwise, getting
I think to really clarify all of the questions you've got, you do need to think about sequencing a wide range of accessions, so that, because if you base it all on one, you don't know how typical that is of the others. We do see in the Australian wild rices that gene flow is quite widespread. We don't see very much geographic structure. So if we take a plant from a long way apart, it doesn't necessarily have a greater genetic distance. So there's a lot of gene movement within the wild populations, but that doesn't mean there's not a lot of diversity. So we see a lot of diversity at each location. It doesn't seem to be geographic. So the main thing that you would need to do is look at a large number of individuals rather than necessarily capture them from a geographic range. But of course, the problem that you have is that you also have a lot of domesticated rice growing in many of those areas. So you've got to be careful that you aren't seeing something that's had gene flow from the domesticated gene pool. And that's one of the advantages that we have in the Australian environment, because in these areas there's been no history of rice cultivation. So we know the wild material is genuinely wild and hasn't got introgression from domesticates. That's sometimes more difficult in areas in Asia where rice has had such a long history of cultivation. But yeah, I certainly agree that you need more sequences to really understand yeah, Olivia's work in Brazil is probably would benefit from having that greater diversity of sequences.
Just among the consensus, that standard sequence of Arizer map alignment, all other species sequences are quite stable. Like, I have found that they are stable across accessions also. The consensus sequence is mostly stable. But in Nivara, this is the sequence which is given, even the genomic locations are also not stable. Actually, that is why you should, with the kind of expertise you are having, kindly go for a sequencing so that the other people working on it can get a, because this is
I think with Nivara we don't have necessarily high quality modern sequences, which could be generated much more readily now, of course, with the latest iterations of the technology. So particularly structural differences, really we need to go back and get de novo genomes, because many of the early genomes were using sativa as a reference, and of course the wild rice might be different in important ways that we just stumble upon unless we go back to first principles and assemble them de novo. But I think that there really does need to be a lot more work on Nivara, because I think it does represent that separate domestication that seems to have happened, and is probably the source of the two. The other chloroplast, there really is only two. All these other variations are nuclear genome.
Yeah. Thank you. So next, Dr. Lotan Kumar Bose, who is a principal scientist working on wild rice since long time in our institute.
Yeah, good afternoon, sir. So really, I got a very good information and from your presentation. So I am actually working on Oryza meridionalis and Oryza australensis. So I have long back collections of Oryza australensis. So I found that particular this Oryza australensis flowers in Indian time, or in India, it flowers afternoon. It starts from 3:30 to 5:30, number one. Number two, Oryza australensis I crossed with diploid and tetraploid sativa, cultivated one. So when I crossed with diploid, then I found that meiotic imbalance in meiosis. And yeah, and in case of tetraploid when I cross, then I got F1s, but I could go for the BC1. But in case of diploid, I could not go for BC1. So settling and all problems are there. And number two, for the meridionalis, it was showing very good for drought tolerance and also grain characters. So I got BC2 and BC3 populations. There is no problem. But australensis, since it flowers afternoon, so the temperature sterility, I think it will be very good effective use for, because it flowers after 30 when the sun ray and all these things are there. So my point is that whether I should go ahead with diploid or tetraploid cultivated for generating or to developing breeding lines that will be utilized in the crop improvement program.
Both sound like interesting options. It's a very important observation you've made there. I think Australiensis is very diverse. It's got lots of very interesting traits that everyone can see now are of value. What I think is remarkable is that people have been able to get genes from Australiensis into domesticated rice, despite the fact that it's such a long way away, in evolutionary terms, 10 million years or more. But it doesn't seem to prevent us with some great difficulty in getting genes across. And my understanding is that in some countries, varieties have now been released that have their disease resistance from Australiensis. So it has proven to be a useful source of novel resistance to leafhopper, for example. I just encourage you to continue working on it. It sounds like a great line of work.
Thank you, sir.
Okay, thank you. So maybe we have another couple of questions. So Dr. Sudha, please.
Congratulations, sir. It was a very learning experience, sir. Thank you for this nice presentation. Sir, my query is regarding can you throw some light upon the aroma of pandanus and the aroma of rice, whether the aroma exists in wild in any of your accessions as well? That is my question, sir.
Yeah, the aroma in pandanus is associated with the same chemistry, exactly the pandan and the rice, of course. We haven't observed any fragrance in any of the wild accessions I've been working with. It's very difficult, of course, to assess at an individual plant or individual grain level. It is true that any mutation that results in loss of function of this gene, which is a 4-amino-butyldehyde dehydrogenase, leads to fragrance. And so increasingly, as we've looked around the world, we do find more and more alleles that lead to fragrance. But I think the vast bulk of the alleles are the same 8 base pair deletion that we found in basmati and jasmine rice in the early experiments. But that more alleles have been found in China, in Japan, in many countries, but they all represent other mutations that lead to loss of function of the gene. They can either disrupt the amino acid sequence, or they can disrupt expression. So they can be promoters there can be mutations in regulatory sequences. But anything you do that disrupts the function of the gene leads to fragrance. But it's very hard to find it in the wild. So it just proves how ingenious the people that originally domesticated wild rice were, that they could actually, the fragrance trait, that they could actually capture this gene that's quite hard to detect at an individual plant level. So nothing in the wild that I've seen.
Okay, thanks a lot, Professor Robert Henry, for your wonderful talk and then obviously very engrossing discussion with our participants. So now it's time for the presidential address by Dr. Dipankar Maiti, Director, National Rice Research Institute.
So thank you, Dr. Kutub. Greetings from National Rice Research Institute of Indian Council of Agricultural Research, India, to one and all. Respected Professor Robert Henry, Professor of Innovation in Agriculture, Center for Crop Science and former Director of Queensland Alliance for Agriculture and Food Innovation, Australia. The August gathering on this virtual platform, officials from ICAR headquarters, directors of ICAR institutes, scientists, faculties of different agricultural universities of India and Australia as well, my colleagues of National Rice Research Institutes and other ICAR institutes, and my colleagues from Australia, ladies and gentlemen. It was really my proud privilege to listen to Professor Henry, an eminent scientist in the field of genomics for improvement of major crops. As we understand from the talk of Professor Henry on genomics of wild and domesticated rice, research on genetic diversity, domestication, and evolution of rice is very much needed to develop improved varieties for more variable and challenging climates, and to ensure food security of a growing human population. Recent progress on genomics and bioinformatics fields have enabled characterization of wild relatives and cultivated species of crop at whole genome level. Sequencing of wild species has revealed genetic diversity in parts of genome that has limited variation due to genetic bottlenecks associated with domestication of crop improvement. The wild species of Arizer are also well recognized source of QTLs and genes for yield and adaptation of biotic and abiotic challenges. Improved understanding of genomics of wild and domesticated species of this globally most important crop requires coordinated efforts to use and analyze rice genomic resources available worldwide for benefit of the humankind. In India, after realizing the first green revolution during 1960s, with the introduction of dwarf gene, yield plateauing was observed during the next decade due to challenge-induced shifts in the pest scenario. Such pest shift scenario were addressed by introduction of new gene from the unexplored gene pool of the domesticated germplasm during 80s, which again faced a climate change effects necessitating the introduction of gene from wild rice to address the evolving stresses. As you know, crop wild relatives are often adapted to extreme environments, and genomic analysis of wild population in contrasting environments may provide insights into how plants adapt to climate variation under natural selection, and this may define options for developing climate resilient rice varieties. Thus, pre-breeding has become part and parcel of crop improvement in all food crops. We are delighted to listen to such a thought-provoking lecture on this very interesting and important field of genomics of wild and domesticated rice, and also a few more crops by Professor Henry. This elaborate talk by Professor Henry has enlightened us, and I believe this will influence the young researchers to think and do further research on different aspects of rice domestication using genomic tools. So I take this opportunity to thank again profusely Professor Robert Henry for deliberating such a resourceful lecture. I look forward to continuing our association in future endeavor and collaboration to address the challenges to food security worldwide. So thank you very much.
Thank you. Thank you, sir, for your wonderful presidential remarks. And Now it's time for vote of thanks. Dr. Somnath Roy, please.
Thank you, Dr. Kutul. I'm feeling honored to get this opportunity to conclude today's special talk on genomics of wild and domesticated rice for celebrating 75 years of India's independence, or Azadi ka Amrit Mohotsav at ICAR National Rice Research Institute, Cuttack, by proposing the vote of thanks. It is my privilege to thank today's guest speaker, Honorable Professor Robert Henry, who is the Professor of Innovation in Agri at Queensland Alliance for Agriculture and Food Innovation, the University of Queensland, Australia, for kindly accepting our invitation and delivering this special talk. Sir, your speech was so thought-provoking. It really made us think. Thank you so much for such a wonderful speech. I'm sure that your speech will inspire us to undertake genomics-driven research on the aspects of domestication and evolution of rice, and also developing climate-resilient rice. Sir, we are grateful to have you as a special guest. I also take this opportunity to express our gratitude to our director, respected Dr. Dipankar Maiti, for presiding over this function and for your constant support and guidance to make this program a success. Thank you so much, sir. I'm thankful to Dr. P.C. Roth, Chairman of the Organizing Committee of Azadi ka Amrit Mohotsav at ICAR-NRRI, for planning and coordinating all the activities. I also thank Dr. Sudhamay Mandal, Member Secretary of this program, for meticulously planning and organizing all the activities and the events for celebration of Azadi ka Amrit Mohotsav. I'm grateful to all heads of divisions, scientists, head of office, senior finance and accounts officer, AAOs, and all other staffs for attending to this special talk and providing necessary support. I also express my gratitude and thanks to all the directors, scientists, researchers, students, staffs of other institutes and KVKs who were attending this event on virtual mode across the country and from Australia as well. I thank all the staffs of Arizer and committee members of Azadi ka Amrit Mohotsav at NRRI for making all necessary arrangements for successfully organizing today's special talk. With this, I stop here. Thank you all once again. Over to you.
Thank you, Dr. Somnath Roy. So I think, Director Sir, should we call it an end?
Yes.
Okay, so before we end, we have a small request to Professor Dr. Robert Henry, because many of us are working on wild relatives, and we might write some clarification email to you. Please reply to us. It would be
please follow up with any questions. Yes.
Yeah,
yes.
thank you.
Thank you very much.
Thank you.
Thank you, Professor.
Okay.
Thank you very much.
Thanks. Bye.
Thank you, Professor. Bye-bye.
Thank you very much.